We are right on time, 8:45, and it's my great pleasure to be chairing this session. We are going to have like three wonderful speakers in 3 different countries, in actually 3 different time zones. And the very first one is actually Tim Connallon, that he's joining us from the future. So he's 8 hours in advance, down under. And so Tim is a lecturer and a head of the biodiversity research group at Monash University. He has a large, let's say, scope of interests. But more recently, he has focused on the genetic basis of the evolution of separate sexes, including zooming in on antagonistic selection and the effect and fate of a structural variance. And so without further ado, so the room is full, is yours. See you.
Okay, thanks, Ricardo. I hope the sound is okay. And so thank you. Thanks, Ricardo, Paul, and Tatiana for inviting me, and thanks for sort of putting up with the remote venue for this talk. So I wanted to talk about one project that we had recently worked on. So Colin Alito and I published this not too long ago. And we haven't done a lot of inversion work, but I thought this context would be a good time to talk about it. So, I probably don't need to give the obligatory 1-slide introduction about inversions being important and contributing a fair bit to conspicuous polymorphism and divergence, but they do. And so what this does is it raises the question about, what are the evolutionary forces that contribute to the spread of an inversion, and either its maintenance polymorphically or its fixation within the population. And this is a really challenging question to address because, a lot of the inversion substitutions that have occurred in the distant past. And it's difficult in even the best of contexts to directly estimate how selection might be acting on a particular genetic variant. In some of these cases, we need to rely on some sort of indirect signal of how process relates to evolutionary pattern. So there's a variety of potential processes that could be important in terms of how inversions might evolve over time. So there could be inversions. Is the sound okay, Ricardo? Yeah, okay. Inversions might have direct fitness effects, or they might have no direct fitness effects at all. So we could imagine scenarios of inversion evolution involving neutrality. We can imagine scenarios in which the breakpoints of the inversions or some other position effects might have, in some rare cases, beneficial effects, and so the inversion could respond directly to positive selection. Or we can imagine that an inversion might cause underdominant effects because it disrupts meiosis. And then there's a variety of potential indirect effects. These are often the focus of studies of inversions. Inversions suppress recombination, and as a result, the genotypes that an inversion happens to capture might allow the inversion to spread because an inversion happens to find itself associated with a good genetic background, at least initially. And so a variety of scenarios could be involved. It could be inversions capturing locally adapted combinations of alleles, and the inversion could spread in a local population because it has a good local genetic background. The inversion could capture epistatic allele combinations that are beneficial in combination and hold them together. You get efficient selection on epistatic combinations. Or it could be that the inversion captures a relatively, or a genetic background that's relatively free of deleterious mutations. And this could be favorable in some contexts. And the challenge here is how do we distinguish between these different processes? So there's an interesting paper that came out, and this paper hasn't been cited a lot. Many of you will probably be familiar with this, but I wasn't familiar with this paper until just a couple of years ago. So Lee Van Balen and Richard Levins had an interesting paper that came out in MNAT several decades ago, in which they proposed that the sizes of inversions might actually carry some signal about the evolutionary processes that were important in driving the inversion spread. And they mostly focused on high frequency polymorphic inversions. And their idea was that large inversions might be good at capturing favorable combinations that are recombining within the base population, but the inversion can capture them and hold them together. So what they came up with was a simple model for what distribution of inversion sizes they might expect to see if there was nothing interesting going on. And this I'm going to refer to as a random breakpoint model. The idea is that inversions might arise if the breakpoints of random inversions are just uniformly distributed across a chromosome arm. They'll follow a characteristic distribution of sizes. And so you can plot this is a plot from their figure. This curve right here is the prediction for what the cumulative distribution of inversion sizes would look like if inversions just form by random breakpoints across the chromosome, and then they just spread subsequently. And then they compared inversion size distributions in a variety of different species. These are common polymorphisms, and they found that clearly this null model isn't a great explanation for the actual inversion size distributions that you see. And there's two potential contributing factors here. So one could be that the process generating new inversions doesn't give a inversion size distribution that matches this. So it could be, mutational causes explain the discrepancy between what you actually see and this null prediction. But another possibility could be selection could have some role in causing a differential spread of inversions that were either relatively large or relatively small. So it could be that selection actually acts on inversion length. And so selection might shape the distribution of inversion sizes that we see. So a more recent study, this is still not that recent, but the idea is that it's hard to separate the mutation from the selection process just by looking at the inversion size distributions, if you don't already know very well what the distribution of inversion sizes among new mutations would be. But one way to potentially get around this is to compare the size distribution of very rare inversions, which presumably would be closer to what the spectrum of sizes for spontaneously arising inversions would be, to very common inversions, either common polymorphic or fixed inversions, which presumably should be shaped by the mutation process as well as selection. And so a paper, 25-ish years ago, sort of found what they found was that for very rare inversions, there were a higher number of very small inversions in that distribution compared to the inversions that become common in the population. So they actually found an enrichment of intermediate sized inversions that reached high frequency compared to the spectrum of sizes for very rare inversions. And so it could be possible that selection is differentially maintaining intermediate sized inversions in this case. But one thing that I wanted to sort of focus on today is what are the predictions? How do different processes of selection on inversion lead to different patterns of inversion size distributions? And so there's different hypotheses out there in the literature. There have been for some time. A couple of these hypotheses were addressed by Van Balen and Levins. So the idea is that if inversions are strictly evolving neutrally, or it's selection on the breakpoint that's going on, we don't necessarily expect any relation between an inversion size and its likelihood of evolutionary success. Alternatively, we might, in different models of selection compared to neutral and direct selection on the breakpoints, we might expect these different scenarios would favor different sizes of inversions. So a recent paper by Cheng and Kirkpatrick sort of emphasized this idea that underdominant inversions probably are more costly if they're larger compared to if they're smaller. So there's more scope for disruption of the meiotic products. And so we might expect that underdominance should favor the disproportionate spread of pretty small inversions. Another scenario is when the inversions capture good allele combinations and hold them together. So this epistatic selection and this local adaptation scenario, these indirect selection scenarios on the inversion, that would potentially favor large inversions, because the larger the inversion, the more likely you can capture alleles at different loci that are recombining in the base population, and you can lock them together with the inversion. So Van Valen and Levins emphasized this hypothesis in their study. And then there's been some studies focusing on the effect of deleterious mutations on the evolutionary dynamics of inversions. And here it's complicated. There are factors about deleterious mutations that are sometimes you might expect them to favor small inversions or large inversions. So Colin and I were interested in trying to pull together different strands of theory and try to come up with some predictions for the size distributions of evolutionarily successful inversions. And the basic model that we want was actually presented in the Van Valen and Levins paper. And so here's a model for the distribution of evolutionarily successful inversions. This function right here, this probability density function Gx, depends on two factors. So it depends on the distribution of new inversion sizes, which is represented by this function f, and it depends on the relation between inversion size, denoted by x, and its probability of fixation. So this function p is the inversion establishment probability or inversion fixation probability as a function of inversion size. So always I'm going to be referring to inversion size using x, and x I'm scaling to be between 0 and 1. So it represents the proportion of the chromosome arm that the inversion spans. So this all applies to paracentric inversions. So x of 0 means that it doesn't span hardly any of the chromosome arm, and x of 1 means that the inversion spans the entire arm. So there's a couple of models that have already been used for the cases of new inversions. So there's that random breakpoint model that I mentioned, and then there's also this exponential model that Cheng and Kirkpatrick used. So in both of those cases, these are plausible models. We don't really know what the underlying distribution is, but there seems to be in either case more small inversions than large inversions. And I'm going to look at both those models when I'm modeling the distribution. When it comes to models for the relation between inversion size and probabilities of fixation, there hasn't been a lot of work. So Cheng and Kirkpatrick looked at one case where the size of the inversion was affected fitness in a linear manner. So what they looked at was a case where larger inversions had a stronger effect in both heterozygous and homozygous individuals for the inversion. So we focused on 4 scenarios of selection. We focused on a scenario where the inversion is neutral. And when I refer to neutrality, what I mean is sort of like the inversion has no effect at all in the absence of deleterious mutation considerations. So of course, when you bring in deleterious mutations to the mix, then there's nothing is really neutral, but neutral is just referring to selection in the absence of deleterious mutation effects. So there's the cartoon. We're considered a case where there's positive selection on the inversion, perhaps due to a position effect. And here's the scenario we would be looking at. We looked at a case of underdominance, and then we looked at a case of local adaptation, where there's an island population receiving migrants, and selection can favor inversions that capture the locally adapted combinations of alleles, shown in black here. So each of these models has different parameters. So the parameters in the local adaptation model involves migration and selection in favor of the locally adapted alleles. And so I'm going to quickly go over the results. First I'm going to focus on what are the basic predictions if we ignore deleterious mutations, and then I'm going to ask what should the effects of deleterious mutations more or less qualitatively do to these predictions. So in the absence of any deleterious mutations, the results aren't particularly surprising. It sort of goes along with the verbal intuition we had before. So the idea here is that for strictly neutral inversions, and for inversions under positive selection due to position effects, there's no relation between inversion length and fixation probability. So the gray line here. For underdominant inversions, the larger the inversion, the lower the probability of success of that inversion. So we see the red line right here. And for the local adaptation scenario, we've got a positive association between the inversion size and its fixation or its establishment probability. Okay. And so if you take into account the size distribution of new inversions entering the population, so this is using the random breakpoint model for the distribution of sizes for new inversions, you get a distribution of sizes that look like this. So for most of the models, we see that there's a bias towards most of the inversions that are evolutionarily successful are small, and there's a long tail of some intermediate and large size inversions. But for the indirect selection scenarios, a selection for local adaptation that the inversion captures and suppresses recombination between the locally adapted alleles, that scenario favors a size distribution with an intermediate mode. Okay, so what happens when we introduce deleterious mutations to the mix here? First thing, and I think this is probably obvious to everybody already, but the nature of the problem, the population genetic problem, is pretty difficult. So if we want to introduce deleterious mutations, we have to think about a multi-locus scenario now, where individual loci are potentially evolving under mutation, selection, and drift, and we could potentially have associations developing between loci as a result of patterns of recombination in the base population, patterns of purifying selection against deleterious alleles, and drift as well, and effective population size. So this raises some logistical issues. One, to fully embrace this complexity, it decreases the analytical tractability of the model, at least for what we could do. And then a second issue that we had is that some of our predictions for fixation probabilities, we expect fixation probabilities for some of these scenarios on the order of 1/n. And so the likelihood of success for some of these events is very, very small, which requires a high degree of replication if we're to approach this by simulation. So what we did to sort of get around this is we used an approximation. So what we assumed that if fitness effects of mutations are at different loci are multiplicative, and we assumed that the population size was sufficiently large, perhaps we can consider the evolutionary dynamics of mutations as largely being deterministic. And if we can make this assumption, then we can model selection on the inversion in a predictable manner. So we can sort of collapse this model down into a model involving really just a single locus, in which we have this selection on the inversion is time dependent. We sort of, this is sort of an open question about how well these approximations will actually work. I expect that they probably will be okay if the population is large enough, but I think there's going to be some population size below which these things will start to fall apart. So as you're looking at the next set of results, you can sort of think about them as something that you should probably trust most when the population sizes are pretty large. That's when they should be okay. Okay, so how do deleterious mutations in general, according to this model, affect the fixation probabilities or the establishment probabilities of inversions? So we can think about there being a cost and a benefit associated with being a large inversion. And the cost of being a large inversion is that you're more likely to carry, or you're more likely to, by chance, capture at least a few or potentially very deleterious mutations. And this is really going to prohibit your potential to contribute to evolutionary change. So larger inversions represent a larger mutational target, and you're more likely to carry enough deleterious mutations to render your evolutionary importance probably not very important. But then again, on the other hand, there's sort of like, if you happen to be a large inversion, and you happen to be pretty free of deleterious mutations, you have a short-term advantage because your load is considerably less than the load of non-inversion bearing chromosomes in the population. And this advantage declines over time. And we can look at these two factors as being benefits of costs and costs of being large inversions. And the balance between these benefits and costs differs between the different scenarios of selection. So for the neutral case, it turns out that if your population size is sufficiently large, these two benefits and costs roughly cancel out with one another. And it will cause inversions sizes to be, or the fixation probability of neutral inversions to be roughly independent of the inversion size. Now, we also find that this large N approximation will break down if as the population gets smaller and smaller. And we also found effect is if the distribution of fitness effects of deleterious mutations also has a big effect on the relation between inversion size and fixation probability for these neutral inversions. So we find if there's a greater distribution, a more skewed distribution of fitness effects of deleterious mutations, it preferentially selects for smaller inversions. So the second thing we looked at was the case of underdominance with deleterious mutations. And we saw here that there was a sort of complex interaction between selection imposed by underdominance and selection due to purifying selection. So here, if the underdominance effect is weak, we find that it behaves kind of like the neutral models, where if anything, you get preferential selection for even smaller inversions with deleterious mutations than without. However, there are some cases where we found that if the load of deleterious mutations on the chromosome was large and the underdominant selection was strong, in some cases you could get a peak of relatively large inversions fixing. And this is because the inversions that are deleterious, free of deleterious mutations, can sometimes drive past the intermediate unstable equilibrium in the underdominance model. And so you get this effect arise sometimes. 5 minutes. Okay, and lastly, we found that, I'll just go over this quickly, in cases where there was some sort of positive selection on the inversion, either whether it was direct positive selection or whether it was selection due to the inversion capturing a good combination of alleles in the context of local adaptation, introducing deleterious mutations into the mix tended to cause the distribution to become smaller than it would be in the absence of the deleterious mutations. So when we take these factors in together, we can see what the effect of the distributions would be under these different models for selection on the inversions and for the different distributions of sizes for new inversions. And we see in both of these cases that intermediate sized inversions are favored when selection for the inversion is due to its recombination suppressing ability for locally adapted alleles. And we find that smaller inversions contribute to evolution when under the other scenarios. And generally what we find is that the introducing deleterious mutations shifts these distributions to the left and will select for small inversions, but nevertheless, you still see an intermediate peak for these local adaptation scenarios, which distinguishes them from the others. So the general predictions from our models, our qualitative predictions is that indirect positive selection should often favor intermediate size inversion distributions, a distinct pattern from the other scenarios of selection. The other scenarios should tend to, if anything, favor smaller inversions relative to larger ones. And there are some cases where we found inversion size distributions within intermediate mode. Some based on cytological data from the past, and this is data from Drosophila in which the X seems to follow this pattern. This is analysis of genome sequence data. We also found that there was different sensitivity to NE for the different models. So underdominant inversion substitutions sizes should decrease with greater NE, so we should see a greater skew towards small inversions when NE is large under the scenario where we got underdominant selection is the most important scenario of selection. For neutral inversions, if anything, we expect that the bigger the NE, the larger the inversion should be on average. And then the other models are a little bit less sensitive to NE, provided selection is relatively strong. And then each of these models is sensitive to the mutation load. With the greater the load for the chromosome, the smaller the inversions we would expect. And so I just wanted to thank my co-author, Colin, who was also on this paper. And then several people made very useful comments during the process we were working on this. And if you want more details, you can check out the papers or you can email me about it. And I think I'm almost out of time, but hopefully there'll be some time for some questions. Thanks.